Hello everyone, my name is Jessica Acosta. I'm a Ph.D. candidate in the monogastric nutrition laboratory of Dr. Hans Stein at the University of Illinois, and today I will share with you the results of one of my experiments where we observed that the nitrogen retention was not different in pigs fed diets supplemented with 65 parts of DL-Methionine or 100 parts of methionine hydroxy analog as a calcium salt. This work was presented at the ASAS Midwest meeting on March 11, 2025. So let's get started. Usually, in a corn soybean meal diet, methionine is the second or third limiting amino acid. Therefore, methionine needs to be supplemented in this diet to reach the requirement of pigs. For this purpose, several sources of methionine can be used. Two of the most common sources are DL-methionine or the methionine hydroxy analog in a liquid form as a free amino acid, or the calcium salt of hydroxy analog of DL-methionine or MHA, as I will refer to it for the rest of the presentation. The main difference among the sources of methionine is in their chemical structure. DL-methionine contains an amino group at the alpha carbon, whereas MHA contains a hydroxyl group bound to the alpha carbon. The differences in the biochemistry between these two sources of methionine may lead to differences in the metabolism and utilization of methionine by the pigs. For protein synthesis, only L-methionine is used directly by the pig. The methionine must be converted to a ketomethionine by the enzyme D-amino acid oxidase that then allow the ketomethionine to be transformed to L-methionine via transamination. MHA provides both D and L isoforms of 2-hydroxy-4-methylthiobutanoic acid or HMTBA, which are also converted to ketomethionine by the enzymes L-hydroxy acid oxidase or D-hydroxy acid dehydrogenase that then allows the ketomethionine to be converted to L-methionine. The enzymes involved in converting HMTBA are present in pigs' livers and kidneys. However, the differences in the metabolism among the methionine sources lead to differences in dose response, which can affect the relative bioavailability observed in pigs. Previous research has evaluated the bioavailability of the methionine sources, and it has been reported that as a free amino acid, MHA has a similar relative bioavailability when compared with DL-methionine. However, other researchers have also reported that the average bioavailability of MHA as a free amino acid or as a calcium salt relative to DL-methionine is only 65%. It has been demonstrated that all genotypes of pig had lower nitrogen retention compared with newer genotypes of pigs, which increased their nitrogen retention by around 60%. Knowing that modern pigs may retain more nitrogen, it is unknown whether pig fed supplemented with DL-methionine or MHA will have the same nitrogen retention. Therefore, an experiment was conducted to test the hypothesis that pigs fed 65 parts of DL-methionine or 100 parts of MHA as a calcium salt will have the same nitrogen retention. Moving on to the materials and methods, I will start with the diets. A basal diet was formulated using corn, soybean meal, field peas, and whey to contain 0.22% of standardized ileal digestible methionine. This diet was below the requirement reported by the NRC by 35%. The basal diet also contained 1.35% of standardized ileal digestible lysine and 2,400 kilocalories per kilogram of net energy. 4 additional diets were formulated by adding both sources of methionine, DL-methionine or MHA, in a low or in a high level. DL-methionine was supplemented at 0.030% as the low dose or 0.090% as a high dose, and MHA was supplemented at 0.046% as the low dose and 0.138% as the high dose. I will present the analyzed composition of the diets in this table. As a quick note, all other amino acids were at the same level among treatments. The only difference was in the analyzed methionine, which was 0.24 in the basal diet, and by the addition of 0.035 or 0.083% of DL-methionine, it reaches the level that were described previously. However, when we analyze the methionine in the diet supplemented with MHA, we observe that the analyzed methionine was the same as in the basal diet. This is because the MHA is not recognized by the standard analysis, and an additional analysis was needed to recover the methionine that is supplemented with MHA. We were able to verify that 0.054 and 0.136% of methionine was added in the diet supplemented with the MHA, and therefore those diets had a ratio of MHA relative to the L-methionine around 65%. All diets were also under the requirement for growing pigs for methionine reported by the NRC. In terms of the animals, we used 30 barrows with an initial body weight of 16.37 kilos that were housed in metabolic crates that allow us for the total collection of feces and urine. We used experimental periods of 13 days with 7 days of adaptation and 4 days of collection. In terms of the experimental design, we used a randomized complete block design because there were two groups of pigs that were allotted to the metabolism crates. But we ended up with 6 replicates per treatment. Data were analyzed using the procedure mix of SAS with the pig as the experimental unit, the diet as fixed effect, and the block as random effect. Orthogonal polynomial contrasts were used to evaluate the linear effect of increasing levels of the methionine sources. Now, moving on to the results. First, I will take some time to set up my slides. In yellow bars, we observed the pigs that were fed the basal diet. In blue bars, we observed the pigs that were fed diet supplemented with DL-methionine, and in orange bars, we observed those pigs that were fed diet supplemented with MHA. Darker colors represent the low doses, whereas lighter colors represent the high doses. When we look at the feed intake among treatments, we observe that the pigs that were fed diets containing DL-methionine and MHA had greater feed intake than those pigs that were fed the basal diet, and only a linear effect was observed in the pigs that were fed DL-methionine. This effect was also observed in terms of the nitrogen intake that was also greater in those pigs that were fed diet supplemented with DL-methionine or MHA compared with those pigs that were fed the basal diet. When we look at the nitrogen excreted through the feces, we observe that the pigs that were fed diet supplemented with DL-methionine or MHA tended to have greater nitrogen in the feces compared with the pigs that were fed the basal diet. Only a linear effect was observed in those pigs that were fed the DL-methionine diet. However, the nitrogen in the feces is highly influenced by the activity of the microbiome, and therefore, to understand the utilization of nitrogen by the pigs, we analyze the nitrogen in urine. We observed that the nitrogen in urine was reduced in those pigs that were fed the diets containing DL-methionine or MHA, and in both sources of methionine, linear effects were observed by reducing the amount of nitrogen when increasing the levels of methionine in the diets. This indicates that by the greater inclusion of DL-methionine or MHA, pigs are able to utilize more nitrogen, and therefore the amino acids are less deaminated, and less deamination results in less nitrogen excreted through the urine. When we look at the apparent total tract digestibility of nitrogen, we did not observe differences among treatments. This was expected because the majority of nitrogen in the diets is coming from the ingredients, and by the supplementation of the diet with DL-methionine or MHA, there is not a huge impact that then will result in greater digestibility of nitrogen. This result was also observed in terms of the nitrogen absorbed by the pigs in grams per day. However, when we look at the grams of nitrogen retained per pigs per day, the nitrogen retained was greater in those pigs that were fed diets supplemented with DL-methionine and MHA, also observing linear effects with both sources of methionine were used. This indicates that the pigs are able to utilize the methionine that is coming from both sources of methionine for protein synthesis. base, and therefore more nitrogen is being retained, and as I mentioned previously, less amino acids are being deaminated and less nitrogen is being excreted. In both sources of methionine, we observe the same lineal effect, which indicates that there is no difference on supplementing 65 parts of DL-Methionine or 100 parts of MHA in terms of the nitrogen retention. This greater utilization of nitrogen by the pigs results in greater final body weight in those pigs that were fed diets containing DL-Methionine or MHA compared with the pigs that were fed the basal diet. With that, we can conclude that feeding 65 parts of DL-Methionine or 100 parts of MHA results in the same final body weight, the same apparent total tract digestibility of nitrogen, and the same nitrogen retention. And this is important because then when we formulate diets, we can use 100 parts of MHA or 65 parts of DL-Methionine, and we will obtain the same results. With that, I would like to thank the Monogastric Nutrition Laboratory and Evonik for the financial support of this experiment. Thank you for listening to this podcast. And if you are interested to learn more about other topics in swine nutrition, feel free to visit our website at nutrition.ansi.illinois.edu.